My name is Logan Everett, and I work at the U.S. Environmental Protection Agency Center for Computational Toxicology and Exposure. My poster is on chemical safety screening using high-throughput transcriptomics. The views expressed are those of the presenter and do not necessarily reflect the views or policies of the U.S. Environmental Protection Agency. US EPA is developing new approach methodologies for chemical safety screening. One such method of interest is high-throughput transcriptomics, or HTTR, which uses low-cost targeted RNA-seq to broadly profile biological responses to chemicals in cultured human cells. Figure 1 on the poster shows the study design that we have developed for performing transcriptomic profiling. This design allows us to profile the concentration-dependent transcriptomic response of a particular cell type in culture to multiple test chemicals, and is intended for an automated laboratory workflow using 384-well plates. Our design includes multiple plate-level controls and reference samples to ensure high reproducibility, and randomizes treatments across replicate plates to reduce technical artifacts. Each set of chemicals on a dose plate is used to generate 3 replicate test plates using independent cell culture batches. The results shown in this poster come from a proof-of-concept pilot study using the MCF7 breast cancer cell line exposed to 42 test chemicals. However, this design can ultimately be scaled to screen thousands of chemicals against multiple cell lines. Figure 2 shows the bioinformatic workflow for processing data from HTTR screening studies, which includes multiple novel QC metrics tailored to the targeted RNA-seq platform and study design. In our pilot study, we applied and compared multiple analysis approaches to characterize the potency and biological response for each test chemical, the results of which are shown in later figures. Figure 3 shows multiple QC metrics from our pilot study and demonstrates that the vast majority of samples have sufficient depth and coverage for reliable analysis. Rare low-quality samples are removed prior to further analysis. Please see the digital poster for further details on each QC metric applied. Our screening protocol includes standard reference chemicals on every test plate. In Figure 4, the transcriptomic response to each reference chemical in our pilot study is compared to gene sets associated with the appropriate molecular target, demonstrating the biological accuracy of transcriptional responses observed in our study design. Figure 5 summarizes results from the 42 test chemicals used in our pilot study. The left-hand panel shows the wide range of transcriptomic responses, represented by the number of differentially expressed genes observed across chemical and concentration space. This demonstrates that overall, our test chemicals vary in terms of both potency and the breadth of the transcriptomic response. An important concept in chemical screening is the point of departure, or POD, which is the lowest concentration at which significant bioactivity is estimated to occur, based on fitting a dose-response model to the observed effects at each tested concentration. This figure shows multiple POD estimates for the 42 test chemicals based on the different transcriptomic analysis approaches summarized in Figure 2. POD estimates are also shown from the ToxCast project, which used a battery of biochemical assays to perform high-throughput screening. Overall concordance between transcriptomic and ToxCast-derived PODs is high for this set of test chemicals, particularly when using the signature aggregation method developed as part of this work. Thus, we conclude that high-throughput transcriptomic profiling of cultured cells represents a reliable and accurate strategy for large-scale chemical safety screening. Thank you for visiting my virtual poster, and please feel free to contact me with any questions.